an Egyptologist. He clandestinely, secretly extracted whatever he could from the mummies and developed this technique of hybridization and showing our genetic lineage. So this is where science merges with fiction actually. Great progress has been done in gene therapy. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. It is not uncommon, at least in foreign countries, that people are testing the fetus for any defective genes, and there are methodologies where they can replace the faulty genes. Now, technology and innovations can be used to save animals from extinction. Artificial intelligence, the cloud, smart cameras are being used to catch poachers and track wildlife populations. Innovations and international collaborations are essential parts of research at present, and I am sure that there will be deliberations on these in this conference. At present, science are real druids, developing new ways to improve our life in every way. With these words, I would like to say what a pleasure for me it is to be in this campus amongst all of you and see my whole all developmental biologists lining up in different capacities and conducting this meeting, and I am their guest actually. I am very proud of them, and I thank organizers. They have always been very kind to me. They haven't forgotten their old professor, and they have given me this opportunity to be here and address you. Thank you very much. I wish the conference all success.
Thank you, ma'am, for giving us an insight to the varied dimensions of science. May I request Professor Sushil Kumar Datta, sir, President, Zoological Society of Orissa, to felicitate our guest of honor, Padma Shri Professor Priyampada Mohanty Hechmani, ma'am.
First time in my life, I am sharing a dais with her.
Thank you, sir and ma'am. May I now invite Organizing Secretary Dr. Gunanidhi Sahu, sir, for the vote of thanks.
Respected all, dignitaries on the dais, of the dais. In fact, all the dignitaries on the dais are my teachers. And of the dais are, all of them are my well-wishers. Organizing an international conference in this department with inviting people from different parts, it's a dream for this department that came true after a long time. This has become possible because of the collective efforts, support of so many people, primary being the participants, the resource persons, the students, and my university administration, and my colleagues. Organizing any conference or that kind of aggregation of scientists, researchers of a particular field has a single objective of developing some or other kind of collaboration among the participants. The number of representatives from the number of institutions present here, and the great teachers, scientists and academicians, the directors, professors, head of the departments from abroad and across India. I believe that definitely some kind of a collaboration will develop out of this. I request each of you to explore all the possible ways by which we will be able to get benefited by some or other kind of activities through collaborations. At the outset, I take this opportunity to thank my Vice Chancellor in absence and the University Administration, who readily permitted us and assured us all kind of support when all the three teachers we met her. Then the sponsors, organizations sponsoring like Science and Engineering Research Board, Government of DST, Government of India. Then the Russia, Zoology Utkal, Utkal University, and the World Bank, Odisha Higher Education Program for Excellence and Equity, all they have supported us financially. So we are grateful to them. I am extremely thankful to all of you. Regarding today's chief guest, because all she suggested us that I will be in a meeting. So please go ahead without wasting a minute even. So in her absence, we thank her. And to our guest of honor, our teacher, rather I feel we have a right to ask our teachers to be with us. Instead of thanking them, I believe that we have a right. And our teachers have permitted us that right. So we are grateful. We are indebted to you and will be like that throughout, ma'am. I believe that whatever we are today, each of us probably because of our teachers, because they saved us, whatever we are today. I am the first Master of Philosophy student of Professor S.K. Datta, so I do have the same right. You will be astonished to know that the 3 international delegates from USA, 2 and 1 from Belgium, they have managed their travel costs themselves. At this stage, when we are looking for small things and looking how to compromise, they didn't bother to spend that much of money to come over to India just with a message or some kind of interaction with them. So no amount of words, sir, Professor Luden and Professor Parajuli, of course of Professor Murugan. Yes, ma'am, please.
Great violation of the procedures, but since I am the teacher, I can tell them what to do. I would like to thank Savita Acharya, the Vice Chancellor. I was hoping that she will be here. I could thank her personally for taking such interest and encouraging this kind of deliberations. I hope you can meet her at some time. And secondly, if my name became well known for research in amphibians with Sushil and Prabhati, all my research on sea turtles, for which I am also accredited with, was done with Gunadidi Sahu. So he is my sea turtle man and these are my amphibian man, because you know, at our age when you are professors, you hardly do any work with your hands. These are the people who made you what you are. So if they want to thank me publicly, I want to also bless them publicly for being in my lab and continue with it. This is an amazing thing. And an anecdote. When we determined that turtle, the temperature dependent sex differentiation in sea turtles, I was going to Vittarkanika, where the largest sea turtle rookery of the world is lakhs, hundreds of thousands come. There, it is the center for crocodile conservation, especially the nasty saltwater crocodile, Crocodilus porosus. So the forest department people told us, Madam, you are doing wonders with this, little turtles. You are making them boys and girls according to your wish. Why don't you do something for me, for us? I said, what do you want? He said, for the last 10 years we have been releasing only females. Because from the natural hatching, only females are coming out. And see what will happen after 15 years, 20 years to this conservation program, there will be no males. So please try to do something. Fortunately, at that time, I was going to an international conference of archaeology and met the expert. He said it is just the trick is reverse. These are really things which I want to share with you, how little things help. In turtles, we found that higher temperature will turn them into girls, and lower temperature will turn them into boys. And the temperature in between, which we call the pivotal temperature, will give us both. And Mother Nature has it very nicely done, because the first nesting is in winter, the second nesting is in summer, towards summer. So both male and female. But you should have just reverse that and put the eggs in the reverse order. So I requested the forest department people who delivered the eggs, which is a surprise, because sea turtle eggs are somewhat like smaller chicken eggs. And these crocodile eggs are like 5 chicken eggs together with large ones. And I had known what temperature they hatch, normal, so I asked them to give me the temperature at which they are hatching in nature. So they brought that temperature to me. So I put 2 groups above that and 2 groups below that. I divided them. And you won't believe, all the ones which are at the higher temperature were boys. So my boys, our boys are doing very well in Vittarkanika actually. Thank you very much.
Ma'am, you at the lighthouse for each of us, showing the ways throughout. That whenever we asked these 3 gentlemen, including Professor Murugan, for any kind of things, they simply told that you need not ask us and go ahead. What do you want from us? So I really, I don't have words how to express my feelings to each of you. My colleagues in this university, they supported me like anything. Around 1 month back, I met Padma Shri Professor Etidas in the Institute of Life Sciences, Bhubaneswar, when during the talk of the Nobel laureate, there I requested, sir, we are proposing to organize an international conference. Then he told, okay, go ahead. I told, really difficult, sir. But I did the flutter thing. And 3 days back, his secretary called me that sir was telling that you have an international conference. So when sir will be going? This is sir beyond imagination. This shows how proves you repeatedly that how you are an institution builder, not only the director of several institutions working with who or anywhere, but you are an institution builder. We are overwhelmed by your presence, sir. Representatives from so many universities, from north to south, and from Kashmir to Kanyakumari, universities and institutions, we are indebted to each of you. My colleagues from different departments, for whatever we have asked, they without any condition supported us. And my students, probably they have done things which they have never done earlier, and I doubt whether they will do it in future, in setting the department and making the department little bit ready for our delegates. For the last 7 to 8 days, they were working almost up to 9 o'clock, 10 o'clock, yesterday up to 1 o'clock in the morning. So my dear students, probably I will remember this throughout my life, being my academic children. I will never forget you in my life. All the 3 teachers who are present here will never forget you. You will always remain in our hearts. And this is a greatest opportunity for each of us to learn so many things, starting from how to cleaning, sweeping to how to excel in research or academics or wherever you are interested. Persons from I'm thankful to the persons from media houses. I thank each and every person, those who have supported us in any kind. I understand that we could not reach to expect the expectations of some of you or maybe most of you. I request each of you to kindly bear this and intimate us. We'll try our level best to manage the things. Tomorrow onwards, we'll have all the deliberations as per the program schedule that has been shared with you right at 10 o'clock. So kindly come in time. Once again, I thank each of you, and I wish that definitely will place on record that yes, something has come out of this. One more thing I wish to place on record, that when we requested Professor Parajuli and Professor Luten that would you like to visit places of interest, historical interest or tourism interest in Odisha, simply they told you please organize meetings with your colleagues or nearby institutions, whoever are there, we just want to interact with them. We are not going to do anywhere. So this is science. Science is nothing like 9 to 5 business or 10 to 5 business. Science is nothing like comfort. Science is nothing like any way we interpret or take our own way. Many of you are the examples. I thank each of you. Let us make it together, make it a grand success. Thank you so much. Namaste and Pranam to everyone.
Thank you, sir, for your warm address. That brings us to the end of this inaugural ceremony, and we will continue with the technical session. Thank you, sir. Also, there is a short announcement. If anyone wants to avail the e-copy of the book of abstracts, you can scan the QR code we have provided outside. Also, whoever wants the hard copy of the book of abstracts, we will provide it on the last day of our conference. Thank you. welcome to the technical sessions. Let's begin with our plenary talk session. May I now invite Professor Pramod Chandra Rath Sir, Professor School of Life Sciences, Jawaharlal Nehru University, New Delhi, to chair this session. Sir, please. Thank you, sir. Let's invite our first plenary speaker, Dr. Walter Leyden, sir, Professor Pharmaceutical and Pharmacological Sciences, KU Leuven, Belgium. His research area, novel bioactive peptides and natural products. He has done his Doctor of Medicine in the year 1981 and Master of Computer Science 1982 from Ghent University, Belgium. He has done his PhD in 1988 from University of California, San Diego for thesis research on nicotinic acetylcholine receptors in the laboratory of Steve Heinemann at the Salk Institute, La Jolla, California. He is a postdoc in the year 1989 in X-ray crystallography at University of California, San Diego in the laboratory of Joseph Crott. To his credits, he has worked in drug discovery and early drug development at Janssen Pharmaceutica, a subsidiary of Johnson & Johnson. In 2001, he co-founded IRIDM, another KU Leuven spin-off focused on peptidomics and G-protein coupled receptors. He has been an independent biotech consultant since 2000 till present, advising government agencies like the Flemish Council for Research Policy BRWP, charities like the Wellcome Trust UK, academic labs like the GIGA in Liège, Belgium, biotech companies like Tevecoma, Complex, and private investors. He has published over 165 papers in international peer-reviewed journals with a H-index of 50. He has co-invented over 30 patents. And today his title of the talk is Natural Products as Potential Novel Anthelmintics. Sir, please.
Okay, I want to thank first of all the organizers for inviting me and for the very generous reception that I've received so far. My talk will be about one aspect of our research. I've decided, given the theme of the conference, to focus on our work on anthelmintics, compounds that combat compounds that combat worm infections in the intestine of humans and animals. My talk will consist of a brief introduction of the topic of intestinal helminths. I will outline our strategy, how we selected plants based on ethnopharmacological knowledge, how we purified the active compounds and identified them, the pharmacological study. I will show you some specific cases of plants where we went through this process and draw at the end some conclusions and talk about future prospects. And to keep time, I will check my phone. Okay, so intestinal parasitic infections are still quite common. About a third of the world population is affected, and still this leads to 200,000 deaths annually. They form part of a much wider set of mostly infectious conditions called neglected tropical diseases because they are more common in tropical countries and oftentimes neglected, certainly by the pharmaceutical industry, but even by academic researchers in the West. In addition to humans, they also cause problems in animals, both companion and farm animals, and therefore the economic burden is actually huge. It's so large that I couldn't find figures. There are only figures available for specific helminthic infections in particular species like in cattle or in sheep. Unfortunately, even though there are effective treatments available, drug treatments, increasingly resistance is encountered, especially in farm animals, which are regularly dewormed. To make matters worse, most of these active drugs actually belong to a relatively small number of therapeutic classes. So if there is resistance to one, there may well be also resistance to other drugs in the same mechanistic class. So there is a real need for new anthelmintic compounds, preferably with a novel mechanism of action. So to illustrate the occurrence, this is two ways to represent the helminthic infections in humans. The top, the top picture shows the prevalence of the infection. This is basically the percentage of the general population that suffers from helminthic infection. And this is another way to represent it. This is the proportion of the global population that's affected. So you see that it is sort of tropical, even though fairly northern countries are also affected, and also southern regions. And India and China actually have the largest number of people affected in absolute terms, in part because they are the most populous nations in the world. Here you see a number of compounds that are used in the therapeutic classes. So there are basically three different therapeutic classes. The first act on tubulins, the next group acts on nicotinic acetylcholine receptors, and the third on GABA and glutamate receptors. You see that shortly after their year of introduction, already the first reports of resistance occurred. And as I said, in one particular class, resistance against one oftentimes means there's also resistance against the other drugs from that same class. Now, we chose to focus on natural products as a source of novel anthelmintics because of a number of reasons. First of all, there is a large body of traditional knowledge of plants that are used to treat helminthic infections in humans, but even in animals. And we start from this knowledge that's oftentimes kept by traditional healers. We did not use self-medication by animals. It's known that some animals, when they're ill, they seek out in nature particular plants that they benefit from, but this we did not use as a point of departure. Because we traditionally used plants, there is already a good idea of their safety and potential side effects. And in some cases, these plants may even be cultivated rather than collected in the wild, which is very useful if you need them in large quantities. Unfortunately, even though many plants are known, the compounds that underpin their biological activity, their pharmacological effects, are oftentimes not known, even though the plants may have been known for centuries, and even less is typically known about their mechanism of action. But we do know that in general these natural products differ from synthetic chemicals in that they have other chemical scaffolds and therefore oftentimes also a different mechanism of action. Peculiar for intestinal parasites is that the active compounds, when they stay in the gut, actually can reach fairly high concentrations for a relatively small dose, because the volume of the gut is fairly limited. And if the active compounds are not taken up from the gut into the body, the systemic side effects are typically quite limited. The strategy we used is that we chose a model organism called Cinerapditis elegans, a model nematode that has been studied extremely well and that offers some advantages when you want to look at mechanism of action. We chose a motility assay using this device. It's called a microtracker. It basically accommodates a Well-to-Well 96 plate, and there is inside there are a number of beams of light, infrared light, that basically, when there is movement in one of the wells, then the light beam may be interrupted, and this allows you to count the number of interruptions, which is a measure of the amount of movement in one of the wells. So what you get out of this instrument is an automated count of the motility over time, and this is per well. So we put some C. elegans in the well, and we have a test compound, and then we see how the motility changes over time. If the worms are paralyzed or killed, then they stop moving, and we can see that with this device. So that's our screening assay. And the starting material is plants. As you can see, we collaborated with a great many different groups. I mentioned their names here, from countries in India, we worked with Dr. Panda here, but also in China, where there's extensive use of plants in traditional Chinese medicine, but also in some folk medicines. We also worked with Yoshibishu from Nepal, some African countries like Rwanda and the Democratic Republic of Congo, Cameroon, Algeria, Cote d'Ivoire, and Kenya. So thanks to all these collaborators, we had access to local plants that had been used traditionally for treating intestinal helmintic infections. Typically, we then make extracts from these plants, and we make extracts in 4 different solvents. Hexane, acetone, ethanol, and water is a common choice, so that we can extract compounds with very different physical-chemical characteristics. And these extracts in different solvents are then tested in the device that I showed you on C. elegans. Now, what we could conclude from this work is that most of the plants that were selected based on ethnopharmacological evidence show activity in C. elegans. So this means that the plants actually were selected on proper grounds. Sometimes we see no activity in our assay. This could be due to the fact that the plant simply is not active against C. elegans, which is a model nematode, not a parasitic nematode, or that we use the concentrations that were too low. We may have lost the synergy, or some compounds may be prodrugs and may have to be converted in vivo before they're active. But we just continue with the most promising plants, and it's not an accident that we find these plants to be active. If we choose other plants, also other medicinal plants that are not used for treating parasitic infections, we typically have a much lower rate of finding active extracts. So the pharmacology definitely provides an enrichment, and it permits us to select promising plants for further studies. The studies we do after the first test for activity is we try to identify which compounds in the extract are responsible for the helmintic effect. To do this, we use a bioassay-guided purification. That means that we follow the activity through all fractions of every separation step. We use chromatographic separations to separate this very complex extract into fractions that are simpler and that can be tested. Each fraction is tested for activity, and we only follow the fractions for further purification in the next step if there is activity in the previous step. We believe this to be superior over phytochemical approaches, so other people, they just try to identify as many compounds as possible in the extract, and then they test these compounds for activity as pure compounds, and this also can lead to the active compounds, but one never knows if these really form the major bioactive compounds or not. Now, the purification is one thing, but identification is oftentimes tricky. The purified compounds, we try to identify them first by mass spectrometry, which is an indirect identification because it only gives us the mass, and sometimes if we fragment it, also knowledge about the fragments of the molecule, but we oftentimes at the end of the mass spectrometry are still faced with several candidates that fit the mass spectra, and then we need to distinguish between those using additional information like elution times on certain columns, the UV visible spectrum, and so on. We can also use NMR to elucidate the structure and identify the compound, but unfortunately this requires several milligrams of pure compound, and this is not always possible from the sources that we have. So I now want to go over a few cases where we went through this procedure that I described. And the first one is work that we did together with Dr. Pander on Oligar and Kaustica. And this is the first step. So we use silica gel cone chromatography to separate large-scale extract of Oligar and Kaustica. And what you see here is the different fractions, and for each fraction we did an activity assay. This is the positive control, levamisole, a well-known anthelmintic, and the solvent control, diamysol. So what you see is that there is one fraction that really stands out and that has almost 100% activity, and in the other fractions we see virtually no activity or very, very slight activity. So we continue with this most active fraction and separate it further by HPLC, and you see that we can resolve a number of different peaks, and the major peaks were all tested individually for activity, and again that you see compared to the positive control, there's one peak that stands out as being the most active peak. These other ones have very little or activity statistically indistinguishable from the solvent control. So this peak then was identified as linoleic acid, both by mass spectrometry and NMR. Here you see the structure of linoleic acid. So it's a fatty acid with two double bonds. And this is actually a fairly simple case. In all the fractions, only one seems to be active. And so in the end, you end up with a single compound from a very complex extract. Of course, this compound has been known for a long time, and also its anthelmintic activity was known. So maybe it's a bit disappointing that you identify a known compound with known activity, but okay, this happens oftentimes with natural product research. Having isolated the active compound, you can do further pharmacological studies like dose response curves and toxicity assays, and we could therefore determine the potency of the compound at IC50, so the concentration where it has 50% of its maximum effect is about 0.4 micromolar, and actually this compares favorably with the reference compound, levamisole, which in fact is IC50 almost 10 times as high. We did not see any cytotoxicity on the human cell lines, and indeed it's known that this compound, which is in fact found as a nutrient in many plants, is not toxic to cells. The second case I want to present is traditionally used medicinal plant in China. The preparation is known as semen Fabitis, so it's the seeds of a plant, Fabitis nil, which is used in China for treating intestinal helminths. And here you see different extracts, different solvents that we prepared. This again is a positive control, and you see that the ethanol and the acetone extract basically are the most active, and this is not uncommon because ethanol and acetone extracts may overlap significantly in their composition. So when we then prepare a large-scale ethanol extract and separate it on silica gel, you can see that most fractions show very little activity except these two fractions. And when these are further separated by HPLC, and then all the fractions are tested, you again see that there's only one area where there's significant activity, and that corresponds to this peak. It's not a single fraction, so it's a somewhat broader peak, and the reason is that in fact this is not a single compound, it is a mixture. So here you can see the general structure of the compounds in this mixture, and here you can see, depending on the positions, the substituents at various positions, you find basically a group of similar compounds consisting of a fatty acid that's modified by a sugar chain via hydroxyl group. So since it's not a single compound and it's a very complex compound, we tried to see if some fragments of it would be active. We cleaved off the sugars by acid hydrolysis, and the activity was lost. So the sugars are essential for the biological activity. We also tried several smaller fragments. So this is forbitic acid. So it doesn't have some of the modifications here, and some other fragments of the forbitic complex, but none of them were active at reasonable concentrations. So it seems like we need the full molecule for activity, and fragments so far have not proven to be useful. We also tested the mechanism of this forbitin, and to do that we used a number of mutants in C. elegans. So these are all the codes of mutant strains of C. elegans that are resistant to one or more of the classical anthelmintics. And we exposed these mutants to the forbitin complex, and what you see is that the forbitin is active on all these mutants except this one. This one is, notice the difference in scale, this one is much more resistant to forbitin. So this suggests that forbitin acts via the target that's mutated in this worm, and this target is a nicotinic acetylcholine receptor. So even though this compound doesn't look anything like nicotine or acetylcholine, it does seem to affect this receptor and therefore act in a way that is similar to some known anthelmintics like levamisole. The third case is another traditional Chinese medicine. It's another seed of a plant called the Torreya grandis. It's a large tree, it's a pine tree, and the nuts of this pine tree are actually sold in China as snacks. So it's a very safe preparation, it's in fact a food. And we use the same strategy. Again, you see that the ethanol, the acetone extracts are the most active, and that when you start to separate the ethanol extract on the silica gel column, this is again a positive control. There is only a single fraction that seems to be active. When you separate that further on HPLC, this is the activity profile. You see that there are two regions of activity. This is again the positive control. And these two regions of activity were further separated by HPLC. And here you see the HPLC peak pattern of those two fractions. And the active compound is, in the first case, this compound, and in the second case is this compound. So it's not the dominant compounds in this fraction, it's fairly minor peaks, suggesting that these are compounds that are not present to high extent in high concentrations in the extract, and therefore they could be missed during, for instance, a phytochemical strategy. So again, we identified these compounds. Both compounds are known. Compound 1 is glycol acetate, and compound 2 is myoganium. Their potency is less than what we found for the previous, compared to the previous compounds, but still in a range that should be therapeutically achievable, certainly in the gut. Here we also tested for activity against the same panel of mutant worms, and in contrast to the previous set of data, what you see that for both compounds, so in gray you have the glycol acetate results, and in black the myogonial results. Basically, all the mutant worms are sensitive to both compounds. And this means that they exert their activity via a different mechanism of action than the traditional anthelmintics. So these are useful as point of departure for obtaining anti-helmintic compounds with a novel mechanism of action. We also showed that there was some synergy between those two compounds, but I have no time to show the data. And the large case study I want to present is a compound from Kenya. It's well known in West Africa. It comes from a tree called Warburgia ugandensis, and I won't show you all the data, but okay, here you see a picture of the leaves and the branches of the tree, and basically we found 3 different compounds. One is again a lipid similar to linoleic acid, and the other 2 compounds are close analogs. One is polygonial. This is the hydroxyl analog of polygonial. We also did some mechanistic studies and found that basically polygonial inhibits mitochondria, and the mitochondria being the source of ATP. Basically, the effect is that the cells are depleted of energy, and therefore the worm probably dies. So these 4 cases illustrate that it is possible to identify using bioassay-guided purification the active compounds in these extracts, and some of these compounds, as I said, were known compounds with known anthelmintic activity. Some were known compounds, but the activity was not known, and some of them had probably mechanism of action similar to known anthelmintics, and some probably have a novel mechanism of action. So in conclusion, we think that ethnopharmacological data can provide useful guidance for the selection of promising plant to discover novel anthelmintics, that bioassay-guided purification guarantees that we can isolate at least the major bioactive compounds, but the identification of these compounds at times is still a challenge. As I mentioned, we often identify known compounds, but in a number of cases, these compounds have not previously been reported to have anthelmintic activity. And again, sometimes we identify compounds with a novel mechanism of action, but in those cases, it will still be quite a challenge to then find out what that mechanism of action is. We are not here at the end of the road. In academic terms, this is oftentimes as far as one can take it. But of course, we would like to see some of this research applied and turned into anthelmintic preparations or drugs. But the road from scientific discovery to such commercial application is quite long and also expensive. There are important regulatory hurdles. One would think that since these plants have been used for such a long time, that it would be easy to get permission to market these preparations or drugs based on them, but that is not at all the case, especially in Western countries where medicinal plants are not commonly used anymore. Although recently some of the regulatory hurdles have been cleared or at least lowered. Another problem is that if you cannot get intellectual property protection. If you cannot get patent protection, then companies are very reluctant to commercialize these findings. And then thirdly, there are also economic hurdles that remain. These plants, when grown locally and collected from the wild by individuals, of course do not cost much, but if you want to scale this up and make them available on a large scale, you have to think about, okay, what is the cost of goods? Are you going to commercialize a purified compound or a crude extract or a fraction of the crude extract? And all of this will, of course, determine the economics of the preparation. It's not all negative. There are also some positive trends in the public acceptance of natural products. In fact, some people prefer using natural products over synthetic chemicals. And also some government policies, at least in the EU and the US, have recently made it a little bit easier to and also favor the application of natural products over synthetic chemicals. So with that, I want to thank you for your attention, and I am happy to answer any questions that you may have.
Hello. So thank you very much, Professor Oscar, for a very exciting talk. And we invite questions. Professor, yeah, please give the microphone if there is one.
I was looking at the distribution worldwide. Uganda is the maximum, you said? It's kind of in Uganda. Yes, that is the maximum occurrence of this disease, or what did you say that? Frequency of the disease.
So I think the question, if I understood it correctly, is about how common intestinal helminths are. So, as I mentioned in the introduction, about 1/3 of the world population suffers from intestinal parasites. So it's a very common problem. It need not be a serious medical problem if the intestinal worms are not numerous. It also depends on which worm. Some worms actually do drain blood. They feed on the blood from the intestinal wall, and they may cause anemia in the longer term. In other cases, they cause problems of pain or interfere with digestion. So there's a range of potential problems that can be caused by intestinal helminths.
Thank you.
Any other questions?
Yeah, yes, please. Question there. Can you please move the microphone here? The first one, yeah. No, the cable.
You must have heard giardia, the protozoan that causes intestinal giardiasis.
Yeah.
So, are there any solutions for giardiasis through the drug discovery that happens through phyto remedies that are known in this region?
Yeah. So, as you point out, Giardia lamblia is another intestinal parasite. We focused on nematode infections, but there is a whole range of parasitic worms, not just nematodes, but also trematodes and cestodes. So there are a number of worms that can be intestinal parasites. There are also other organisms that can be intestinal parasites. So it's not limited to worms. and many of them pose similar challenges to helmintic infections because, again, they're not that common in Western countries, and therefore they don't get the attention of pharmaceutical companies. Now, for some intestinal worms, the traditional healers can make a diagnosis. Some of these worms appear in the stool, so you can make a visual diagnosis, and even people that don't have any modern medicine can come to a relatively conclusive diagnosis that intestinal worms are present and may be causing the symptoms that the patient complains of. For Giardia lamblia, that's not the case. So for traditional medicine, it becomes a little bit more difficult. But I still think that there is potential in natural products. But one needs, in order to do this research, one needs good bioassays, and these are not always easy to obtain. So a bioassay needs to be at the same time easy, inexpensive, and rapid throughput, but be realistic enough so that if you find activity in your bioassay, it will most likely transfer to the in vivo activity. And this is a problem with a lot of modern research. When I started working at Janssen Pharmaceutica, subsidiary of Johnson & Johnson in Beers in Belgium, they screened for intestinal parasites in a dog model. So they had dogs, real live dogs, that were actually captured off the street and that were brought to the company. And they were experimentally infested with worms, and then they were fed drugs, candidate drugs. And then they looked in the stool to see if the parasites were dead or if the eggs would not hatch. And this, of course, is a low throughput assay. So we can only test maybe one compound a week in one dog, because you want to be sure if you test the next compound that the first one has been completely eliminated. So this system allows us to test in 24 hours. We can easily test 100 different extracts or fractions or compounds. But of course, this is a model nematode, not a parasitic nematode. But I didn't show it. But for at least one of the plants, we also tested them on real parasites, Coperia, which we obtained from our colleagues at Ghent University from the parasitology department. And they provide us with the parasites, and we had to adapt the settings of the equipment a little bit. But we could also get the same assay to work on these parasites. I must say, I don't know for Giardia lamblia if there is a convenient bioassay that we could use. But if there is, then I think there is potential for using natural products and pharmacological tools. Although, as I said, I don't think the diagnosis of Giardia is possible without some laboratory testing and a microscope. So we have to go by clinical signs of the disease if you want to tap into traditional knowledge, or just use plants that are used traditionally for treating infections by related organisms, and then hope that the activity will transfer, that activity will be broader than just one target organism.
So thank you very much, Dr. Walton. Now I request the organizers to maybe organize the presentation, please. So we are running short of time. So what has been instructed by the organizers is we organize a discussion at the end of the session. Okay, please keep reading with your questions. And
May I now request Professor Pramod Chandra Rath sir to present a memento to Dr. Walter Luton sir. Now let's welcome our second plenary speaker, Professor Megha N. Parajuli, Professor, Faculty Fellow, and Texas A&M Research Fellow. Research area: cattle entomology, ecological approaches to pest management, integrated pest management, multidisciplinary cropping system, production sustainability. Academic record: MSc, PhD, and MS. Achievements: he is leader in cattle entomology and cropping system research. He is the founding editor-in-chief of Global Journal of Agriculture and Allied Science. He has co-edited a book on principles and practices of food security. He has received the Texas A&M Vice Chancellor's Award in Research Excellence, Faculty Fellow Award. He has been awarded Regents Fellow Award, Texas A&M Vice Chancellor's Award in Team Research, Entomological Society of America Southwestern Branch IBM Excellence Award, and USDA Southern Region IBM Hall of Fame Award. And title of the talk is Integrated Pest Management and Cropping System Research in Texas, Meeting Stakeholder Expectation. So please.
Namaskar, everyone. This is a rare opportunity for me to be part of this conference, and the rarity comes from the situation that I've been already engaged collaboratively with several agricultural universities in India. I've been in the US for 33 years, with Texas A&M for 29 years, and I come to India, because of COVID, it was all messed up, otherwise every year, every other year. But never have had chance to have this type of linkage with state universities such as yours. So I hope today's this presentation will provide some basis for long-term opportunities for collaboration between this university, between this department and our institution in the United States. First of all, I am a little bit embarrassed for the high profile introduction. I am not what is presented here. I guess in the website we have to put all the awards and all that sort of thing. But here I am a research scientist. I stand in front of you to present my long tenure of agricultural research and what I have done in my own laboratory and what I have learned working with farmers, cotton farmers in the United States, and what we have from a systems approach, cropping systems approach, what we have been able to deliver to our producers. So that's what I'm trying to share with this group. And I know that entomology is not really a zoology, but it's a part of the bigger biology, we are within the same umbrella, so I hope the presentation that I deliver here today will have some relevance to students, young scientists, and even the senior scientists for possible collaboration moving forward. Just to give a snapshot of Texas, this is the United States map, and Texas is the southern state, and the map, the highlighted Texas map is not blown up, it's the same size. Just to impress you that Texas is the largest of the 48 contiguous US has 50 states, Hawaii and Alaska being outside the mainland, so Texas is the largest within the mainland. So it's pretty different. It acts like its own country because the enormity of the everything, the agriculture enterprise, industry, and all those kinds of things. A little bit of my background. I'm native of Nepal. I grew up on a small farm, and there on the right side is my home place, still a village in Nepal, and did my bachelor's in ag economics from YS Parmar University in Zoology, India. There in 1987, went back to Nepal and did a little bit of teaching role at agriculture university there for 3 years, and went to University of Wisconsin-Madison, did my master's in 91, PhD in 94 in storage insects, and moved to Texas A&M in June 13, 1994. Ever since, I've been with Texas A&M University System, working in cotton entomology and cropping systems program. So just within Texas A&M, I first started as a postdoc fellow for 2 years, 1990 to 96, and then moved on to become a permanent scientist and cotton entomology program specialist in Vernon, Texas. We have 13 research centers all across the state, and so I was being in charge of cotton entomology in that area, working in various commodities, cotton, wheat, sorghum, canola, and rangeland system. Then moved in 2001 to my current position as cotton entomology program leader. But that station covers the entire state because of the enormity of the cropping system that I'll show you in just a second, where I lead one of the components, that's the entomology component of the overall cropping systems team. We have agronomy, soil science, plant path, economics, and irrigation engineer. We develop a system that is useful for our producer in that region. I'll kind of run you through. I have several slides. I have over 100 slides, but don't worry, I'll try to kind of push it through. All right? Because since I came all the way here from that part, so I thought I'll give you the maximum I can give within the time allotted to me. And I'll share this slide here, so you are free to use this. This is not my personal thing. It's all property of science and humanity. All right? So Texas Cotton IBM, I'll do a little bit of history, livelihood, and technological advancement. You can see this map, this slide should be all the story of my lifetime as an agriculturist. You can see on the top left corner, we are talking about technological advancement, innovation. Look at there from there, now to the lower right hand corner, we have all precision planting. We can plant seed right to where the moisture is in the soil. So even in the field, so we call precision planter, cotton planter. So wherever the moisture variation exists in the soil, so when the tractor driver drives, and then it senses the moisture, and right there at the interface of moisture, that's where it drops the seed. So when people ask, okay, what's the depth of the cotton planting? We don't know. It's where the moisture is. So that precise work we are doing. So we're coming from that to here. That's the story of agricultural scientists. Talking about cotton, I am a cotton specialist. Cotton was a livelihood in Indians up until 8,000 years ago. And we have in the US, it's only 200 years. Cotton has been the fabric of life in the US for only over 200 years, but it has been, cotton has been both from the spiritual standpoint and from agricultural standpoint, it has been around here from Indus Valley since 1,000 years ago. So talking about the technological advancement and the role of science, look at here, some genius guy kind of tethered the plow to a couple of oxes here. That was the science. At that time, that was the genius idea. That was 5,000 years ago. But the beauty of that is that technology stayed for years and years until early 1900s. We are doing the same thing basically. The plow was a little high-tech, but the mechanism was the same thing. So we are moving much forward. And in early 1900s, that's actually my research farm right now in High Plains of Texas, in Lubbock. So mules were the motive force. We were using mules only 200 years ago in Texas. But 20th century combustion engine and tractors brought us a big revolution. It was a game changer. Things changed. Look at here. We have even within the technology, look at the tractor on the top left versus what we have today. So that's the kind of the advancement in technology has been really, it's much the steeper slope now than ever before, and it's continuing. Moving on to that, first fully mechanized cotton production in the United States occurred in 1944, where when I said fully mechanized, planting, harvesting, baling, ginning, everything was done by machine. First one was only 78 years ago. It's not long time ago. But since that time to now, you can see what advancement we have been doing. Biotechnologically, one little seed of cotton has so many things. You have herbicide tolerance, plant pathological resistance, entomology, insect resistance trait, who knows what. One little seed has like 10 different things that scientists could engineer and put it inside. So that's the kind of technology we are talking about. That's the kind of innovation we are talking about. So today's technologically advanced cotton production system gives us such a uniform, it's not a Photoshop crop. I mean, of course I put this together, but it's the real field. This is my own test plot. I took a photo from my phone. So you can see the uniformity and homogeneity of the crops, the height and the size of the bolls and fruit, uniformity in maturity of crop and all those kinds of things. So that's the technological advancement we are talking about. And on the top photograph you can see, again I'm trying to remind myself where we came from and where we are today, and that can tell us where we would be or where our generation would be years from now. Texas, cotton, and IBM, three things I'll try to run quickly through. And Texas High Plains is a cotton country. It has nothing but cotton in the whole the area. We have 1.7 million hectares within 150 kilometer radius. Look at that. So that's the world's largest contiguous cotton patch. But we are a semi-arid region. We get only one annual rainfall of 40 centimeters. But fortunately, and the reason people ask me why you have so much cotton in that area, because nothing else can be grown with 40 centimeters of rainfall, right? So you have no choice. But cotton is an amazing crop. If you have a little bit of moisture and it germinates, it can produce some bolls, some leaf. Not a great thing, but it can. But corn you can do. During tasking stage, if there is no moisture, cotton cannot produce any harvest. So that's why we are the largest contiguous cotton patch in the world. But we have serious issues with shortage of water because it's semi-arid, and we have windy storms like this. These are like we call hoop, and they come almost every year, and it shatters young seedlings, and seedlings can't establish for a month or two until they have good root system. Once the root system is really strong, then it can shoot up. So we have the crop season is very short. Then by November or late October we get snow or frost. So within that narrow window we have to produce crops. So that's the challenge we have. We get this kind of thing, sandblast, hailstorm. I mean, we have every natural disasters that you can, as agricultural workers or farmers can face occurs in Lubbock, Texas, or that area. But still, we do amazing things. You will see shortly. And one, from a scientific standpoint, in the cotton history, this guy, H.A. Maka, he was a farmer in 1926, November 1926 on Thanksgiving Day. That was the historic sandstorm. We have never had such a storm since then. And he was about to harvest crop from the following day, but then next day he went to the farm and everything was blown away by that storm, except he found one single plant. That's the actual picture of that. It's been revolving around until today, and that was by accident finding the storm-proof cotton mutant. So science is sometimes, you don't need to be a genius scientist to find this kind of technological advancement. Yeah, he didn't know what it was, so he was concerned, hey, this is all I got left. And the agriculture said, oh, that is a gold mine here, give me that. So that's the seed. Today, we are giving the benefit of that kind of natural disaster. So that's why we always tell farmers that farmers are ahead of our scientific knowledge that they give us to work on. So we really salute our farmers. we are better scientists today because we have farmers on the other side. So, and Texas farmers are early adopters. Whatever technology we develop, they are trying to, they are early adopters and they take challenges. So look at these are all my own test plots. So they use like heavy equipment, whether the herbicide sprayers or insecticides or tank mixing of herbicide and insecticides and so they can play with anything we ask them to do for. So that allows us to do kind of interesting research. And I'm not going to read all these, but I'll show you the entire cotton industry, cotton enterprise and IBM in Texas and in the United States, mostly in Texas, was driven by this individual insect called boll weevil, Anthrax grandis Bollman. Boll weevils entered the United States from Mexico in 1892. Since then, there was, farmers were producing cotton and so forth, and there was not a major insect problem. But when this entered, it really changed the whole dynamics of cotton production system, and 120-year problem existed. So it took us 120 years to get rid of that insect, and I'll run that through. It's an amazing history. You can see how the IPM has developed during invasion of notorious insects such as this, and after we eradicated that. I'll run it through that, and it would have caused 22 billion in damages by this insect. Okay, so Texas IPM is much influenced by this insect during both invasion and eradication. So this is the border of US and Mexico, Rio Grande Valley, 1892. It was the beginning of the 120 plus year war with one insect. And look at this. Initially, farmers had nothing with which to fight the weevils. So they came and the farmers had to watch. There were no insecticides, no any mechanism to combat that notorious insect. Yield losses were 50 to 80%. Look at the situation there. So it started in 1892. Look at the little green dot there. That's when it started. And watch how the insect moves every year. And it will take probably a minute or so, and it covers the entire United States cotton belt. All the black line covering, that's the only cotton production region. And the boll weevil goes all over, and you can see 1922 way on the top right, it reaches over there. So it took 30 years to overtake the entire US cotton region by one insect, and nobody could do anything. They let the weevil go and overtake the entire region. But that really brought us all kinds of research tools and opportunities. Problems are the mother of opportunities. So if there are no problems, then people we don't seek opportunities. So we had soon all the entomologists came to help. Texas usually is a rich state, so it could afford all kinds of entomologists, behavioral entomologists, insecticide taxonomists, and toxicologists, and all those kinds of people. And I'm going to run this quickly. So they develop cultural control methods, source season concept, and so forth. Mechanical control. First, they had like a rudimentary plow, horse-drawn plow to remove the weeds and so forth, to reduce the pressure of weevils. But mechanized harvest equipment and so forth down the road. So again, there was technological advancement on everything, cultural control, mechanical control, insecticides. 1917 we had calcium arsenate, DDT, on, and 1994 we had neonicotinoids, and which are very selective and very specific insecticides that we are using. So there is again the growth in technological advancement front. And just few slides just to show the kind of insecticide application. And again, technology here, technological advancement. This is how our Texas farmers spread insecticides. Little advanced technology here. Look at this gentleman. He's having a lot better opportunity than the previous slide. And even better here. Aerial dusting started in 20s and 30s. Industry relied heavily on insecticide. Really, there was a little bit of cultural control, mechanical control, but insecticide was the major combat boll weevil. Still, they were not being able to really overcome the major disaster that boll weevil brought to the cotton industry. But soon, mid-1950s to 70s, environment, resistance, resurgence, all those things came into play, safety and environmental concerns. And 1962, we had Silent Spring, written by Rachel Carson, that stimulated everybody to talk about environmental and health and all those kinds of things. So environmental movement really began in the US during that time, and you can see 1959 in Hilgardia, Eastern Adolf published Integrated Pest Management. But notice that 1959, IPM terminology was coined, but our farmers, hundreds of years ago, they were doing IPM, but they didn't say that it was IPM, but there was no other way of doing the business other than IPM. IPM, we are just kind of making that more scientifically prudent and more efficient system, but this is not a new thing. Our farmers were doing already. So we started developing scouting and extension IPM programs were developed in the US, growth of private consulting firm because there was so much pressure on farmers, and integrated pest management really took the root. And that time, total population management thought was developed by Edward Ripley in 1950. That led to the initiation of boll weevil eradication in 1983. So farmers, planners, policymakers all thought that we need to eradicate this weevil, otherwise the cotton industry will collapse from the US. So they started all kinds of methods, and I'll run through a few slides and see how we reverse the situation. So you can see all the blue here, dark blue is the pre-eradication. Those are all covered by, inundated by boll weevils. They were established. And active eradication, there will be red boxes, and then completed, they will turn into green. Just watch here. Because it started from Texas and went to the north and all the way to Virginia, we started to eradicate from there and push it back. We did. We were successful because it was a small area, no problem. And it started California and North Carolina, South Carolina, and continued doing it. It took a little while, but you can see, start seeing greening things, meaning that we are pushing weevils back. And quickly I'll run through that. 97, 98, and a whole bunch of activities during around the middle of the southern United States, Texas included, 2001, 2, and it's basically remaining one in the central part, 2005, 6, look at it, 8, 9, 10, by 2012. Actually, by 2013, we officially declared that United States got rid of weevil, except that little thing there, red, where boll weevil first began. That is a very typical place in South Texas that it's not part of Mexico, but you can't get rid of it. Otherwise, I mean, they inundate because it's just next to the Rio Grande River. So instead of fighting that, we quarantine that. There is a strong quarantine facility. We don't let weevil push to the north. So it took exactly 30 years to push it back. That took 30 years to inundate the whole country. But we feel pretty good. Since 2013, we have not had any invasion of reinfestation of weevil in that area. That was quite a success story. We have 2.3 billion, I think 2012, some economists estimated 2.3 billion economic impact of boll weevil in Texas. And as far as we know, there is only one world's monument to a horribly destructive insect. There is a monument. And I don't have time to tell the story, but I'll be very brief. It's not because boll weevil is such a horrible insect, that's why it got the monument. But because boll weevil destroyed all the crops in Mississippi, Alabama, that area, so they had no choice but to plant peanuts. And peanut actually gave us much more profit, so they were blessing boll weevil to coming and damaging the cotton so they would have better crops. So they honored this destructive insect and have monument there. There you go. So now we are in the post eradication phase. So we, at that time, 1996, we had BT, first available transgenic BT crop, and immediately farmers started adopting, but because of the boll weevil, the BT adoption was hindered because boll weevil won't have any effect by BT. So as soon as the boll weevil started being eradicated from various states, BT adoption increased to near 100%, 90 to 100%. So, and by mid 2000, they started stacking. Here is the history here. You can see we started in, I don't know my pointer, but you can see there. So Cry1ASU, one gene, and then 2003 and 4, 3, 4 until 2014, you got like a double gene stack that gave much better efficacy. And 2014 to 2017, now we have three gene stack. But a lot of people don't know, actually only BIP3A, we are basically a single gene system now because the other two genes don't really work. Even though it's a 3 gene stack, really BIP3A is the only one giving us a control. In Texas, it gives near 100% control. Other states, they have to spray maybe once or so, even with the white stripe or twinkling plus or BIP3A. So in Texas, you can see 1988 all the way to 2020, we used to spray like 4, 5, even 8 times a season. So down to about half spray per season. Tremendous advancement in the pest management system. We have like a quantitatively, we consider that we have reduced insecticide use by 72% in cotton. Similarly with the heliotine, you can see we used to trap 800 moths per trap per week back in 2000 to 2006. Now the last 4 or 5 years, you can see it's about 200 moths per trap per week. The entire population, moth population dynamics also has changed. So a few points on Texas cotton IPM following transgenic crops and boll weevil eradication. We have two things going. We got rid of boll weevil, we have better system, and we have transgenic crop that could take care of most of the heliotines. Largely Helicobacter jaya, here Armigera, which is basically similar thing anyway, and Helicobacter Heliotis virescens. Those two were controlled pretty nicely. So we have preventative pest management tactics, seed treatment, transgenic, those things we use. IPM resistance management of secondary pests are de-emphasized. Great opportunities for enhancing IPM now because we got rid of the major pests, so we can have more ecological approaches of pest management. And that's my area of research the last 10, 20 years. And strong movement away from field-specific IPM. We used to control, we used to deal with the one specific field. Now we look at the more regional basis. Maybe one territory or maybe a state. So we look at more of a landscape level management rather than field by field management. And I'm toward the end of my presentation trying to see, okay, I'm doing okay, looks like. I will give you a few examples of what's my work. I was not there 30 years ago. I was there 30 years ago, but the time of presented work 30 years prior to my 30 years. So using those and how that work has been related to my work here. So I'll give you a few examples here. So one, we are looking at now we have a transgenic system and boll weevil eradicator from the system. We have opportunity to work on insect-plant interaction, plant compensation. So a large part of my work has been how we can utilize plants' inherent capacity to compensate the insect damage or weather-induced damage. Because cotton is basically we grow annually, but it's a perennial crop. So it has the capacity to have deep root system, and it is an indeterminate plant. If you give moisture, nutrient, and time, it will keep growing, right? So I want to take advantage of that inherent capacity of plant and then see how we can manipulate insect behavior and insect damage. So that's the threshold study, seed treatment, and so forth. And again, I'm not going to give data or anything like that. So that's the kind of area of research I've been doing the last several years. And any in the group who be interested in that type of research. That's why I'm trying to kind of show you here what type of research that I'm doing. And I'll give you one detailed example toward the end. And the other is cotton clearwater as one of the mirid, it's a mirid, and soil nitrogen. So looking at different nitrogen level, how it's a tritrophic type understanding here, examination here. So the different nitrogen level will have different plant phenological stages, plant architecture, plant nutrient like a leaf nitrogen is different because there is more uptake and so forth, and all kind of interaction, and how that affects insect feeding and how that affects plants' compensatory ability. Again, idea is how we can manipulate the plant's compensatory ability to harness our ability to manage insects without utilizing insecticides. Third, it is also miridi, like TIASRST, and look at here on the slide on the right side, you can see late season, if they remove some small fruits from the plant terminal, actually it's better, they thin out the small fruits which will not be harvestable fruits anyway. Actually you will not only, I say no effect, but over time actually it increases the fiber quality of cotton because it can mature better. It doesn't have to invest its energy on unnecessary small fruits which are going to be discarded anyway. So those kinds of insect plant interaction studies that I've been doing. And last portion I'm going to show you a little bit of here is we have, as I show you, thrips, clearhoppers, and lycos. Those are the three main bugs, main insects in our system. So the thrips and clearhoppers are what we call is pre-flowering or it's a pre-fruit development stage. That's the more critical stage. So what we want to do is, thrips is pestiferous only for the first 4 weeks. After the seedling emerges from the ground, next 4 weeks, that seedling is susceptible to thrips. After that, plant can tolerate. So you can have any number of thrips, it doesn't affect. Then, next 3 weeks, that insect becomes key player. After 3 weeks, then that insect also is no longer a pest. In fact, it is a predator because it starts feeding on bollworm eggs. So we are trying to utilize that behavior of insects and how, when that becomes pestiferous, when it becomes predator, when it becomes non-important, those kinds of things, and trying to utilize in the system and develop the best management strategy for production sustainability. How farmer can have the best sustainable cotton production system for his cropping system, cropping enterprise scenario. So I had 3 levels of water: dry land, depth irrigation, and full irrigation. Full irrigation, we could achieve about 90% evapotranspiration replenishment. We will never do 100% ET replenishment because of our weather situation. And depth irrigation, we gave about 30% of ET. So we stressed, and then we looked at, how then we said, how about in those 3 situations we have thrips, single infestation, clearhoppers, single infestation, and the third is clearhoppers and clearhoppers sequentially infesting both. When there is only one insect appearing in farmer's field, in those 3 water levels, does he or she need to treat? How about only clearhopper, when both? Which one? So there were 12 different combinations. So we developed more like a management strategy. Look at here. So and then quickly I'll give you the thrips stage. So we release thrips and we have to generate data. And then clearhopper. So there are like a single thrips release, single clearhopper release. So that's the stage, 3 weeks, 4 weeks after seedling stage. That's the size of the cotton you can see on the right side. So that is about 4 or 5 week old cotton plant. Look at it, how small. So because number one, by breeding we are producing a smaller plant, so we don't have to invest that much nitrogen. Because the plant you need to put more nitrogen. Nitrogen is too expensive. So our breeders bred very short stature plant, easier to harvest crop and so forth. So looking at the data here, so see the dry land and full irrigation. Leftmost bar is the control, and the second to left, the second bar is the thrips amount. So thrips reduced yield significantly, kg per hectare, 900 on dry land you could get 900 kg per hectare linked yield, but 200 kg reduced in dry land. Similarly, full irrigation is reduced significantly, but depth irrigation there was not much difference. Cotton flyover is not a significant yield reducer by itself. If thrips don't show up in the field, only flyover did show up, then they will not reduce. Look at the third bar versus first bar. See, so no significant effect. Full irrigation there is a little bit of dip, but no. So flyover by itself won't be reducing yield. So we advise farmers that you don't need to spray for flyover if you didn't have any thrips. What if you had thrips as well as flyover sequentially? You can see dry land, no effect, but you can see the arrow. If you have a little more water given, third of water given, it reduces. But if you give more water, it reduces further. Why? Plants are becoming more succulent. You know, the flyover can breed more, have more population. So the higher input system actually is not very good from the flyover pest management point of view. So sequential infestation of thrips and cotton flyover loss about 20% on dry land and high input system, but in depth irrigation about 10%. So still it was economically detrimental to our growth. So then we went further and did all the revenue and variable cost calculation, lint price, seed price, strip margin, ginning, irrigation, all those things for each of the 5 years. I'll go straight to the data here, and these are only gross margins. We haven't done the detail, fine net profit and so forth. But look at here, thrips reduce gross margin across all 3 water levels. Look at here, thrips, the control versus thrips. Dry land, significantly reduced. Second one, depth irrigation, and the third one. So you can see the big circle, within big circle. All 3 water levels, thrips are the key. You saw the numbers already. That translated to the gross returns. How about flyover? Cotton flyover reduced gross returns. Remember that in the high water there was no per deep, but you can see here $224 per hectare loss. That was significant. But sequential, you saw already the number of insects and the damage that reflected big time over here. Over $900 gross margins on control versus what? Less than 600 on dry land, 600, about 500 for thrips and flyover. So, 450, the numbers are here. So 300, 160 and 450. So there was significant yield loss, even though the key point here is flyovers alone may not be causing significant yield loss, but if flyover shows up after thrips damage, they will really do a significant yield loss. And so dry land and full irrigation systems are quite vulnerable for sequential infestation. So the key here is integrated cropping system with crop budget and enterprise analysis is needed for farmers to really know where to put their money in the pest management. I'll just have a few slides to kind of run through just to kind of capture that. So our relationship with clientele, how we do these researches, clientele group is at the center. I am mostly related to cotton, but there are other commodities, livestock, sorghum also. So we work with the clientele, work directly with Texas A&M Agri-Life Research, Extension, County Program, ag consultants, ag industry representatives, and producer organizations. They all work closely with our clientele. So our idea here is we want to cultivate, enhance the synergistic partnership amongst these constituents to deliver the product to our clientele. And again, I don't expect any of you reading this, but after all this, our producers asked us to do some molecular type work, and here project title CRISPR-Cas9 genome editing of cotton gel genes for agricultural बच्चे हैं हेल्थ वर्क आज भी जो है अर्टिकली रेजिस्टेंट. They wanted us to find, because we have only one of the three statins, only last in is useful anyway. So they said, okay, why don't you work on something, because what if this last gene fails, then we don't have transgenic cotton. So we are partnering with all the orange lines on the input. You can see Texas A&M, Texas Tech, Texas Cotton stakeholders, crop consultants, Plains Cotton Growers, South Texas Cotton and Seed Association, Seed Source Genetics Inc. So all of those partners, we collaborated together to seek money from USDA, and even our relationship with growers, the Plains Cotton Growers, they write a letter to USDA saying, okay, we need funding to do this research because we have 1.7 million hectares of cotton here. So that's how we get support, and that's the expectation of our growers from us as well. To conclude, bovine eradication has been a great success story that led to the possibility of integrated pest management system for us. Currently available cotton weed technology may need supplemental insecticide, and the resistance management will continue to be an important aspect of our IPM program. So that's the last project, USDA project, that our producers supported. Will be dealing with this. What's the alternative? What's the supplemental tool here? Ecological approaches to pest management now drives the Texas cotton IPM. We are not insecticide heavy anymore. It's ecological management. How we can manipulate our crop compensation, water management, soil management, and so forth. Stakeholders are an integral part of our university research extension programs, and technological advancements must synchronize with stakeholder buy-in for social transformation. With that, thank you very much for the opportunity. And I got that from Google. I don't know whether that's correct. Dhanyavad.
Thank you very much, Dr. Mega, for a very interesting talk, but we'll keep the questions towards the end. And now it's time to felicitate Professor Mega, please. I request, no problem. So I request Professor to please felicitate Dr. Mega for his wonderful talk.
Thank you very much. Appreciate it.
Now I would like to invite Padma Shri Professor Aditya Prashant Das, Vice Chancellor of Asian Institute of Public Health University, ex-Vice Chancellor of Central University of Tamil Nadu, ex-advisor of United Nations World Health Organization. He pursued his PhD and DSc degrees in Zoology at Utkal University, Bhubaneswar, Odisha. In his area of research, he established Anopheles annualis as an important vector in rural areas and identification of telomerase activity in gametocytes of Plasmodium falciparum. He published more than 250 papers with high citation index, H index, and I-10 index. He is the highest producer in Plasmodium falciparum research in India during the last 10 years, and one of his papers on malaria was found to be the second most cited one in the last 100 years of malaria research in India, which is among the top 10 papers according to the Indian Council of Medical Research 100-year celebration reports. He ranked among top 10 authors in the area of immunology and microbiology and medicine by Elsevier in their Scopus database, in addition to many more laurels. He started his career as a scientist in ICMR, served as director for 11 years in ICMR and Deputy Government of India. He was formerly the director of Institute of Life Sciences, Bhubaneswar, Director of National Institute of Malaria Research, New Delhi, Director of National Institute for Research on Tribal Health, Jabalpur, Director of National Institute for Implementation Research on Non-Communicable Diseases, Jodhpur, Director of Center for Research in Medical Entomology, Madurai. He also occupied the Distinguished Scientist Chair at the Institute of Life Sciences, So let's welcome him for his talk on the topic Control Neglected Tropical Diseases to Achieve Sustainable Development Goals.
A very good afternoon to all of you. At the outset, let me express my gratitude to the organizers for inviting me to this place to interact with you a little while. And when I saw the topic, I thought I was speaking on control and eliminate neglected tropical diseases to achieve sustainable development goals. As you know, the sustainable development goals are targeted to be achieved by 2030, and there are 17 goals. The Sustainable Development Goals originated from the Millennium Development Goals that continued from 2000 to 2015. So, all the 17 Sustainable Development Goals are revolving around three things: gender, equity, and human rights. And to achieve these things, we have to eliminate the neglected tropical diseases, because there are certain tropical diseases which I am going to tell you. The neglected tropical diseases affects the poorest of the poor, affect the marginalized community. And I am basically a zoologist. I did my PhD honors in zoology, MSc in zoology, PhD in zoology, DSc in zoology. I am not a medical man, but I have been working on these subjects from 1978. My work is related to societal development. So, how this is going to happen? How neglected tropical diseases? What are the neglected tropical diseases? The name indicates that these are the group of diseases which are confined to a particular geographical location in the tropics, the tropical countries. So, these are the diseases categorized as the neglected tropical diseases, and these are found in several countries in Africa, Asia, Latin America, and these are popularly called as NTDs. They are especially common in tropical areas where people do not have access to clean water or safe ways to dispose the human waste. I have categorized into three. One is diseases targeted for eradication, that cures and tuberculosis, and the diseases targeted for elimination as a public health problem. These are diseases. If I tell this, I cannot read because of I have limited time. And these are the tropical diseases, neglected tropical diseases targeted for control or prevention. Out of these diseases, there is a group of communicable diseases which thrive in improvised settings and affect more than 1 billion people worldwide. So these diseases not only survive and spread in the condition of poverty, they also perpetuate the poverty of the affected community. Now, these are in India, they are the previous slide said the neglected tropical diseases is categorized by the World Health Organization globally, but these are I'm very happy that the first presentation, the first lecture was on intestinal parasites. With WHO, we call it as soil transmitted helminthiasis, which has also been targeted for control or prevention. There is no prevention but control. But when you come to India, India has limited diseases. These are two diseases, tuberculosis and EOS. These are targeted for eradication, but I am very happy to say that India has eradicated tuberculosis way back in 2000. And And remember, for eradication of dracunculosis, India did not need any vaccine, did not need any medicine, did not need a seizure to cut the DNA like this to control dracunculosis. But by improving the environmental situation, the dracunculosis was eradicated. And yours was eradicated very recently, 2 years back. And globally, there are only 40,000 cases now. For all these diseases in blue, sorry, are targeted for elimination as a public health problem. And these are the diseases in India targeted for control or prevention. And 195 countries are signatories to for this achieving the Sustainable Development Goals by 2030. By 2030, and you see all these related to, as I said, gender, equity, and human rights. When I say human rights, it covers everything. You tell me, is it not human right to sleep without a mosquito bite? I think this is a human right to sleep with a mosquito bite. Similarly, these are all these things are related to that, but one is related to climate change, and others are related to sustainable development and all these things. So when I said elimination as a problem and elimination, what are the definition? Definition is eradication means permanent reduction to zero of the worldwide incidence of a specific pathogen with no risk of reintroduction. That means eradicated completely. Elimination means interruption of transmission, reduction to the zero level of the incidence of infection caused by a specific pathogen in a defined geographical area with minimal risk of reintroduction. But elimination of the public health problem means eliminate the measurable targets set by the World Health Organization in relation to specific disease, so that there is no more threat as a public health problem. Control means reduction of the disease incidence, prevalence, morbidity, and even mortality in certain cases of it to a locally acceptable level. So these are the United Nations definitions for these things. Now, on 28 January 2021, the Limited Tropical Disease Roadmap was launched. There are 3 fundamental shifts: programmatic action, cross-cutting approaches, and country ownership. Each country should take the ownership of controlling the neglected tropical diseases to guide implementation of NTD activities during this decade. I'll tell you, let's see you what forward. Thank you. You'll have time on this. Okay, let's skip this one. So this is complemented by a set of companion documents that is what by 2030 the roadmap was set, and the World Health Organization declared that 30th January of every year should be observed at the World NTD Day, which was approved by the 74th World Health Assembly. But to see the target by 2030, what they said is that 90% fewer people will require the intervention of NTD. I will be coming about the technological developments later, but now I am giving one idea about that. And 75% fewer NTD, that is 100 countries having eliminated at least one NTD by 2030, and till now 2 NTDs are eradicated. Now, cross-cutting targets are 70% integrated treatment coverage should be there, and 40 number of countries that adopt implement integrated skill-related tropical disease strategies. 90% share of the people at risk protected against catastrophic out of the pocket health expenditure, including disease, to achieve the
Sustainable Development Goal 3.8, 100% access to at least basic water supply, sanitation, and hygiene, etc. in areas. Whereas if you see in 2019, 1.74 billion required interventions against STDs, down from 2.1 billion in 2010. This was the success already achieved. Means 2010, there are 2.19 billion people who wanted intervention for neglected tropical diseases. It has come down to 1.47 billion by 2019, and 1 billion people benefited from at least one NDD intervention. Now the progress highlights, you will see annually over 1 billion people benefited from at least one NDD intervention that is shown in the previous slide. 105 countries require prevention chemotherapy. 81 countries implemented large-scale treatment. 649 million people no longer require MDA at all. For the coverage MDA, I am going to tell you in my later slides, and global burden of visceral dysgenesis, which we call as Kala-Azar. And many of the daily entities significant. Now, on the glass again, you want to see 43 countries or territories and areas have eliminated at least one neglected tropical disease till now. In 2019, Mexico, so this is 2018, or when to take 2019, Mexico, Kiribati and Yemen, they have also Mexico eliminated rabies, in 2020, Malawi eliminated hepatitis, Togo, human African tuberculosis, Myanmar, trachoma, Gambia, trachoma, and Portugal, human African tuberculosis. So we are progressing in a better way in controlling or achieving the elimination or control or eradication of the neglected tropical diseases to achieve the sustainable development goals. If you see the leprosy, look at this graph. This graph should go like this. What is the design increases some countries. So this is what we say that leprosy, there is a new case detection, and currently global trade is 1.5% per year. This is the target is being down to less than 1% for 10,000 population. But leprosy very related problem. Now I'll give you two examples because there is no time. I have said this is a targeted for eradication, this is a targeted for elimination as critical problem, this is a targeted for control. Eradication is not a problem, India has already achieved. Now let's go for elimination and control. I will take one example for elimination, how technology has, and another example for control. So for elimination, I take the example of TIASRST. This is a disease which I was looking after globally, when I was in World Health Organization, looking after retroborn and all neglected tropical diseases, and TIASRST is one of the important diseases there. So here you know TIASRST is caused by nematode parasite, by 3 nematode parasites, and transmitted by different mosquitoes. I will come to that. So now you could see TIASRST. What are the oldest and the most debilitating neglected tropical diseases caused by nematode parasite living in the human lymphatic system? That is why it is called as TIASRST, and transmitted by mosquitoes like Curiosity mosquitoes and other mosquitoes. And some places like Nicobar, Aedes mosquitoes also. It possesses several social and economic burdens of the individual, the family, and of the affected countries. And there are 3 types of federal parasites: Mucilla baratrophi, Lucia malai, and Lucia tibular. They all stay in lymphatic node, so they are called as lymphatic parasites. Mucilla baratrophi is most prevalent and common in Southeast Asia region, including India, transmitted by Mucilla baratrophi, transmitted to the incubators. Asmonia transmits Lucia tiburi, Lucia malai, and all mosquitoes, Anopheles, transmit clearly. So see the importance of zoology. The parasite is involved and human being is involved and transmitted by mosquito vectors. Then this is the life cycle of the parasite. Lack of time. The human being, mosquito takes malaria and develops to the first stage larvae, second stage larvae, third stage larvae. Then the infected mosquito will bite the healthy human being. It goes to become the fourth stage larvae and become adult. It will produce microvilli in separate blood and again taken by animals. So this is a cycle. So once upon a time, in the world of lymphatic analysis, little appreciation of the burden, inadequate means of diagnosis, and inadequate tools for treatment were available. Inadequate strategies to control the infection, little hope or anticipation that things could change and LA can be eliminated as a public health problem. Now, thanks to research and development in 1990, and here comes the technological developments. Now, what happened? I can tell you. But see, when we have lymphatic analysis, the classical treatment is DEC, diphenhydramine citrate. This is the chemical name. It is available in Panoside or whatever. If your user is more than 7%, the doctor will prescribe 14 days of Panoside. Now this 14 days treatment for lymphatic analysis, how it has come. When I joined WHO in 2009 February, even this start, I tried to search the literature which clinical trial has proved that 14 days of treatment of by DEC can cure lymphatic filariasis. Trust me, I could not find any literature, in great literature, in published literature, nowhere. I went to many people to discuss. I have complete freedom. Finally, I went to NIH Bethesda. There was a filariasis group. That group, Pat Lamy, he referred me to another person, Eddie Quotation. And I went to him, an old man. He told me how this treatment has come. How this treatment has come by DEC? Now there is an island, very high endemic for. There is no clinical trial. There is an island, very heavily endemic for lymphatic analysis. A group of laborers were taken by a ship from that island to the mainland, which is 3 kilometers. So what to do? The administration declared you administer DEC to them every day 1 dose till they reach the mainland. So they were given DEC every day 3 milligram per kg weight 1 dose after food. And it took 14 days to reach the mainland. After they reached the mainland, they went to work, they stayed there, established their colonies there. After few years, there is no evidence of lymphatic analysis transmission. So if you are thought for treatment will cure. And that is the clinical trial. Now can you think of that? So anyway, from that it started. Then in 1990, basically 2 zoologists, they started working on this. They proved by mid-1990s that a single dose of DEC can give the equal effect of 14 days of DEC. Then people thought that yes, this disease can be eliminated. There was a mega project in India, and they, from the data, mathematical modeling was done by the London School, and it was mathematical modeling suggested that if you give DEC once in a year to everyone in the endemic population, only 1 dose, for 6 years, with 80% coverage, you will eliminate lymphatic filariasis at the public health. Then the global lymphatic filariasis elimination program has launched. See the strength of scientific innovations. And now, I take when I was working as an auditor scientist in the Zoology Research Center, I got a big project to study the lymphatic filariasis in this region, Khordha area. Khordha area, every house I had visited, 4-5 persons I got saw to be built by the particulates. Not very good. It was very pathetic. But now you go, how do you find this person? And because of this, and in Southeast Asia region, Thailand, Maldives, and Sri Lanka, they eliminated living currency as a public property. And Thailand is one country which is not been to fuel by the national of doing this one to this society, to the people of Thailand. Now, coming to this lymphatic currencies in India, lymphatic currencies are endemic in 270 districts, and in most of the districts, the strategy of elimination is bring down microcarrier density to less than 1% so that it cannot be transmitted to another person. Because when a mosquito takes the dog with microcarrier, if microcarrier numbers are more, mosquito will die. If microcarrier number is much less, hardly any of them will develop resistance. So the proper microcarrier for high transmission is 20 number, not 20 number per mosquito if it takes. But if you bring down to 1% of microcarrier in human blood, mosquitoes will take either 1 microcarrier, 2 microcarrier, which will never hit the resistance. So this was the idea. From that, now you see. Now, out of 198.7 billion people infected in 2005 has come down to 51.4 billion in 2019, indicating a 7.4% reduction. This is the strength of the technology developed in 1990s. Now, coming to another basis, that was all the analysis. Now coming to another disease, I said to see which is calculated control or prevention. That's the diseases. If you see the significant arbovirus causing human illness in Southeast Asia are the general arboviruses, the dengue, Zika, Zika, etc., etc. Yellow fever is transmitted. But not found in India. There are 4 dengue virus serotypes. That is, you have dengue 1, 2, 3, 4 serotypes. Each serotype has also genotypes. One has 3 genotypes, 2 has 2, like that. So these 2, you can see here, transmitted by ABC vector. Like this, it is vectors. The sylvatic species, it still loves pressure. It can breed only when nature is there. It breeds in these places. And if you control Aedes, you control chikungunya, you control dengue, you control Zika, you control yellow fever in India. And see that I said there are 4 serotypes of these viruses. And when you see how these 4 serotypes spread all over the world, now this is the serotype 4. It came from this place, I think. This place, Thailand, Indonesia, etc. And they said, for everything there is an epicenter. You have tsunami, there is an epicenter. Earthquake, there is an epicenter. And the epicenter intensity is very high, then it spreads and becomes weakest. The epicenter for dengue is Thailand and Indonesia. So dengue 4 came here, and then dengue 1 has got from Indian subcontinent to other countries, and dengue 4 again it has come from Indian subcontinent to other countries, and then this is dengue 3, and gradually all serotypes has spread throughout India. And today you see all serotypes are available everywhere in the world. So from pandemic, dengue when came It was pandemic. You recently, all of us have seen the greatest pandemic of the human society, COVID-19. It has become pandemic to 2020, 2021, and now we are living with COVID-19, but in an endemic or not pandemic. Similarly, dengue came as a pandemic, but mortality was 18%. But today mortality is less than 8%, and it has become pandemic with all serotypes available everywhere. So now to see 1991 in India, these are the pockets you have dengue. Now 1996, when you see how it has been, 1921, you can see the map, 26, and by 2013, almost every state is endemic or dengue. So this is, there are many dengue difference between the borders, doesn't need passport, doesn't difference in border reach. You can see TIASRST-2023 of dengue. Then this is what I was taking a team of experts to Thailand interior area to show them the transmission biology of dengue. And this is a organized a big workshop worldwide to explain how dengue can be prevented and controlled. And there are cross-cutting activities, and this is a very interesting thing. I was because I was looking after dengue in World Organization has technical advisory groups, and I was arranging the technical advisory group once in 2012 in Bali, Indonesia, because in Southeast Asia region, Indonesia is the country which contributes the highest number of dengue cases, and Bali state which contributes the highest number of dengue cases in Indonesia. So I organized here. When we were going to Bali, I was going with my wife, my secretary, and Sunil Shetty, who is in family, was also going with us. We did not talk to each other, but while coming back, incidentally he was also coming with us. We were in the business circle now, and my secretary, but I will tell you very different. Is there anybody from Bangladesh? Is there anybody from Bangladesh? Okay, I have no personal things. When I was working in World Organization, I had many Bangladesh friends, colleagues. When we talk to them in Hindi, they say, oh, we don't understand Hindi, we talk English. But when I went from official to Bangladesh, there is a good hotel which is called Sundan Bangla, where the Japanese people we used to stay. And there in the English Saturday night, there are English songs. And I saw most of the people, they come and sing the Hindi songs also. Okay, if they don't understand how you come. But they, similarly, my secretary was from Bangladesh, Mr. Mohammed. He said, I don't understand Hindi. At least speak English. Okay. But when he saw Sunil Shetty, he ran to him, started speaking in Hindi. Sir, my boss is going, my boss is looking after dengue, and he has come here to organize a dengue meeting. The moment dengue was, Sunil Shetty had, it was 2012, 2 months back, TIASRST died, he ran to me, Sunil Shetty. That will take, I have a big NGO, take whatever amount you want, please control dengue in Bombay. I don't say that the doesn't accept money from you. We have so this is the seriousness of dengue. Now, climate change. You know, all vector diseases are climate diseases. Even the epidemic tropical diseases are climate sensitive. We all know that climate change is happening. We cannot say we can avoid that one. So, because the especially vector diseases, the vectors are animals, they cannot regulate their body temperature. They will always take the environmental temperature. And the pathogens can grow in a particular temperature, not below that, not after that. Therefore, global warming has a tremendous impact on vector diseases. And we know this is the impact, this is different vector, but we know the climate change is happening. And especially cold nights and warm nights, this is a neglected aspect of climate change. We generally ignore that. So there are high pillars of sustainable development goals. Number one of equity and human rights, then prosperity, nurture fulfilling of prosperous lives in harmony with nature. Then peace, foster peaceful societies, then partnership, and also planet, protect the environment, human climate change and environment. These are the 5 pillars of the sustainable development goals. Now, I give you another. See, there is a review of which country has progressed how much in achieving the sustainable development goals. See, India, 120. In Bangladesh, above India. Myanmar and Nepal, they are also above India. Maldives with 500,000 population is much above India. So we published that paper in Lancet. But I am giving this lecture here because finally at the end I have to give a message to the universities and colleges. What message will come? Now, how can the universities contribute for the achievement of the sustainable development goals? You know that there is an association of Indian universities. They have meetings every year. 2022 they had meeting in Mysore, and they discussed everything about sustainable. I am just telling you with a briefing in ourselves because of lack of time. And they discussed about everything about the education of governments and etc. and everything. Then they saw when India is 120 number serial number in achieving the sustainable development goals, they had a Mysore declaration. The Mysore declaration says that was adopted in the 96th meeting of the Association of Indian Universities with the National Action Conference conducted in Mysore in March 2022. The basis of the Indian University resolved that the university would draw strategies for achieving their targets for the realization of the sustainable development goals. The PCs have resolved to commit themselves for the realization of the sustainable development goals and align university education, research, innovation, and governance towards achieving the goals and targets set out to reach 17 sustainable development goals. The last one is very important. The Mysore Declaration is a landmark resolve to the university of the universities in India to enable India to achieve sustainable development goals and accomplishments and take India to 20 from 120 in 2022. This was a message for everything. I thank you very much. I can't ask all Thank you very much.
So, we can thank you very much, Professor Das, for a very exciting lecture and also a very important message for all of us. So, it's time to congratulate and show our respect for Professor Das. Although time is very demanding, but we can have few questions. Please move the microphone. Yes, sir.
Excellent talk. I am from Calcutta University, Department of Zoology. Department of Zoology, I am currently serving head of the department. My name is Ena Rai Banerjee from Calcutta University. a great talk, and we definitely need to cut across disciplines to solve problems of society. So, in one of your last slides, where you show that India is ranking below other countries, what are the criteria for scoring?
There are 17 goals, as I said. Okay, 17 goals. We have criteria, we have targets, we have achievements. So that scoring, I did not have time to explain to you. You say that scoring, which has come to which level, which has come to which level, scoring will automatically come to computer. No human being will give that. So accordingly, it is coming to automatically.
I do get that, but often scores are very nuanced. So in order to understand better, that was a question, but it is beyond the scope of just a question answer session at a conference. So maybe we can discuss offline. Yeah. No, you could probably just, okay, we will take the discussion offline. How to get more information on it. Okay, thank you.
Hello. Yes, a very good afternoon. I am Professor Sharwani from Assam Central University. Currently, yes, teacher. Yes. I am from Department of Life Science. I am head of the department and Dean of Life Sciences. So there was listening to your talk, it just came to me like I was just touching the tip of the iceberg, and lots to learn and lots to know. My special interest being, this field, but my research orientation is something different, although I work in environmental health. My question is, with the increased global warming, what is the impact really on this vector-borne diseases, and what work has been done so far in India, and what is the scenario in Northeast India, because I came from Northeast India. So especially the mosquitoes, if I Google it, I don't find even the variety of mosquitoes which are available, and not much work has been done. So please, if you can highlight.
Mosquitoes and vectors are the insects and predators. All insects are phototropic animals. They cannot regulate their body temperature. They have to take 100 temperature. If their 100 temperature is 40 degrees, their body temperature is 40 degrees. For example, malaria. Malaria parasite can develop in mosquitoes if the temperature is more than 14 degrees and less than 30 degrees, 32 degrees in humidity. 22 degrees temperature, maximum growth. But the temperature, body temperature goes beyond 35 degrees, was temperature, and mosquito body temperature goes to 35 degrees. Even if we take malaria parasite from the human being, the parasite should not be able to develop to reach the infected stage to infect another person. So this is gene shortage. At the result, what will happen? The transmission within those different states will change. For example, we always say in Odisha, different states, Assam and other states, malaria is perennial, because every month you get malaria transmitted in those areas. But suppose the temperature increased by 4 degrees, the transmission in windows from 12 months will decrease to 10 months in Odisha, will decrease to 8 months in Assam, will decrease to 11 months in Meghalaya. Like that, we have a you can Google, type in Google Scholars, Wikipedia, see all the papers, select climate research papers from there and get it. I published that paper. I give an example. See, whether the international from malaria research, again, I have time and answer this question, and this is the important. When I was analyzing the temperature of India, I saw the South Himalayan area. We are Uttarakhand, what is the hill station? Uttarakhand, Utkal, and above Nainital, to go above towards Himalayas. In the foothills of the Himalayas, there is no malaria. Even when malaria was rampant in India, people were dying like flies in India in pre-independence India. Because the temperature was very less. Temperature was never going beyond 40-50 degrees to encourage the development of the malaria parasite in coast. Then we saw the recent temperature. It was in 2000, no, it was in 2006 or 7. I saw the recent temperature, and that temperature there was an increase of 4.5 degrees. Then they sent a team, go there, see what is the situation. They came back with four malaria parasites, 4 patients with malaria parasites, both falciparum virus, lot of anomalies prevailing which transmits malaria because of the increase in 4.5 degrees generally. So this is its state, and climate change is again another topic which I next few minutes or something like that. Okay, and same thing with Delhi. Delhi particular colors are less
Thank you, sir. Now we should stop here. So let me extend my heartfelt thanks to Professor A.B. Das for his wonderful presentation, and at the same time, I would like to extend our 2 speakers before Professor Das, Professor Linton, Professor Parajay from USA. So now this time we should break. And before we break, I thank our chairperson of this session, Professor P.C. Utkal, for nicely organizing this. So we will be back, we are a little late now, so we will be back by 3:15 after lunch. Here, we'll start the next section. Thank you very much. Well, there is an announcement.